Good. Good. Good afternoon, everyone. It's a pleasure to welcome all of you here. Thank you for braving it through what was quite a dramatic morning. You would have noticed that we felt the effects not only outside, but inside our building here as well. So we were a bit worried that the afternoon would continue in the same vein, but fortunately not. It seems to be clearing up, and you are here, and that's the important thing. My name is Christoph Poe. I'm the Senior Program Manager at STIAS. I am welcoming you on behalf of the Director of STIAS, Edward Chirumira, who unfortunately has taken a bit ill and was told to go home and rest, which is always good advice. But he sends his regrets and his very special regards to everyone, especially to our introducer and our speaker today. I also want to welcome all those who joined us online. Thank you. It's wonderful to know that there's always a virtual presence to our lectures here as well, and you're all very much valued. I have just one announcement, and that's to say that today is our third lecture this semester of the STIAS lecture series, and we'll have a fourth lecture in the middle of May, 15th of May, when our fellow Alex Eze will present a lecture on his work dealing with the future of population in low and middle income countries. So we look forward to that. But before that, we look forward to today. So let me introduce Professor Ben Lewis. First, Ben has kindly agreed to introduce the speaker, Daniel. So Ben is Associate Professor of Molecular Physiology at Stellenbosch University, also currently Chair of the department. And he leads a project called the Neuro Research Group in that department, which is led for about 15 years now, almost. So Ben's research focuses on understanding how autophagy, a process where cells break down and kind of recycle their own components, affects cell death in brain diseases like neurodegeneration and cancer. And their aim is to measure and influence this process more precisely to better control when and how cells die. Ben has published over 70 journal articles. They've garnered over 9,000 citations. Google Scholar tells us he has an H-index of 29. These are all the kind of numbers that one wishes for when you start your career, I'm sure. The work that his group does and that he does is very much collaborative, involving experts in electronic engineering, computational biology, and they've developed a device that can detect autophagy activity as well as advanced 3D software tools that use virtual reality to analyze cells with very high precision. So very much fitting work to be the introducer for Daniel's work today. And without further ado, it's my pleasure to welcome Ben.
Right, thank you, Christoph, and warm welcome. Good afternoon. It's so nice to see you all here, some familiar faces. And of course, this is about our guest speaker, so I'm very, very excited to introduce this topic. So how can I start? So let me start the following. All humans originate from a single cell that, during embryogenic development, differentiates and multiplies into more than 30 trillion cells, which spontaneously organize into functional tissues and organs of astonishing complexity. Although all cells in the human body carry the same genetic information as the first few cells, our ability to regenerate lost or damaged tissue is severely limited. While some animals can regenerate entire limb structures, humans primarily heal very limited through scarring, a process that often results in impaired function. Lost limbs and organs do not regrow. However, by combining cells biological factors supporting biomaterials, we can now grow structures that resemble tissues and organs in the lab. Using advanced 3D bioprinting technologies, we can in fact recreate well-defined and more complex tissue-like architectures than ever before. By engineering human tissues in the lab, we, our presenter, aim to accelerate drug delivery, drug discovery, and develop personalized treatments that pave the future forward for regenerative therapies. So with that, colleagues, it's my great pleasure to introduce our speaker, Prof. Daniel Aili. He is Professor of Molecular Physics in the Division of Biophysics and Bioengineering at Linköping University in Sweden. He has spent postdoctoral fellowships at the Imperial College London and Nanyang Technical University in Singapore, both by chance I know also very well, and was recruited to Sweden, where he established the Laboratory of Molecular Materials. His interdisciplinary research focuses on bioresponsive materials for biomedicine and bioengineering. He has published extensively, about 100 peer-reviewed papers, authored several book chapters, and holds over 8 patents. So, given the complexity, the urgency of the diseases that we are facing in our society, this kind of work is really at the forefront of translation. And hence, it's a great pleasure to introduce Daniel, and we look very much forward to your presentation. Thank you so much.
Okay, thank you for that very kind introduction, Ben. And thank to you, all of you that have come here despite the rain. And also to all of you that are attending online. I would also like to extend my gratitude and my thanks to the STIAS team that has made my stay here at STIAS such an amazing time. And also, of course, to my fellow STIAS Fellows. This has been not only intellectually very rewarding these last couple of months, but also very fun. So really a highlight in my academic journey so far. Thank you. With that, we dig into this topic. And this funny little looking creature, it's an axolot. It's a freshwater salamander. You can find this, if you're lucky, in some biotopes in and around Mexico City. They're unfortunately on the verge of extinction. But in research laboratories around the world, you can find them in quite large numbers. And the reason for this is that these creatures have an amazing capability that we humans lack. They can heal by regeneration. And that means essentially if they lose tissues or an organ or even an entire limb, that piece of the body will regrow and look more or less the same as the original tissues or the limb. They can even regrow parts of their brains, which is pretty amazing. And humans cannot do this. I guess most of you, or all of you, have had a wound in your skin. And when you look closely, what happens with the skin? I mean, it typically heals after a couple of weeks, right? But when you look closely, you can see that this new formed tissue is not identical to what you had there before. And this is something we refer to as a scar. So it's a slightly different tissue. It has a different tissue architecture and different function. And if we lose a limb or an organ, it typically don't regrow. Some tissues can, like liver tissue, but limbs don't. And in most cases, if you have a wound in the skin, you can survive, it heals, and you can live with it. But if we have an injury to, for example, our heart after a myocardial infarction, you will get scar tissue that impairs the contraction of the heart, right? And that leads to loss of function, and this can be really problematic. Likewise, if you have an injury to your central nervous system, you get scar tissue, and this can also be a complete loss of function. And the reason why we have scarring, that's of course evolutionary. It's been advantageous for us as humans to heal quickly, to restore at least barrier function to prevent us from infections. And thanks to the rapid and wonderful development of new technologies in medicine, we can live with a certain extent of loss of tissue function and loss of organ function. We can replace limbs with prosthesis. We can use medical devices, such as implants, to restore some functions. And we can even transplant certain tissues and organs. The patients has to be on medications for the rest of their life to prevent immune reaction, but it saves the life of patients around the globe. So why can we not regenerate? This is a question that we don't have an answer to yet. What we do know is that the blueprint is there. We have the DNA that codes for the entire body in each and every cell that carries a nuclei. It's the same DNA that was there in the first cell after the fertilization of the egg. And actually, during early embryonic development, humans can regenerate, but this function is lost as the embryo develops further into a fully grown human. But there is a notion that if we can harvest cells, why can't we, in the lab at least, create conditions where we can start to grow something that looks like and behaves like tissues? Because this would be tremendously useful, not only for regenerative purposes, but also in order to have tissues, human tissues, that we can use in drug development, for example, or in fundamental biomedical research. And the research field that deals with these kind of questions is called tissue engineering. And it's a fairly straightforward concept. And it's based on the concept that if we can take cells and combine these with biomaterials and techniques for essentially putting these together, we should be able to make tissues in the lab. And I will go through this a little bit and talk a little bit how this could work and what we in our lab, how we approach this problem. So starting with cells, that's the fundamental building blocks of our bodies, right? We humans consist of approximately 30 trillion cells. That's a fairly huge number. It's about 100 times more than there are stars in our galaxy. And starting with one single cell, as the embryo develops, these cells gradually start to specialize, and we have in the adult human roughly 200 different cell types, as what our current knowledge at least tells us. And for example, we have epithelial cells, we have connective tissue cells, muscle cells, nervous system cells, blood and immune cells, reproductive cells, and a certain amount of stem and progenitor cells, which can recreate some of these more specialized cells. So in order to engineer tissues, what we need to do, we need access to these kinds of cells, right? And the most straightforward way then is take a sample from a person, essentially. We can extract cells. We refer to these as primary cells. And in principle, we can take any cell type and try to grow them in the lab. But you can imagine it's a bit more challenging to extract neurons from the brain. It would be pretty invasive, right, procedure. So this has hampered the development of tissue engineering. But in 2006, Yamanaka and his colleagues developed a pretty revolutionary technology that enabled us to essentially take any cells, for example, a skin cell, and reprogram it back to the state where essentially it reminds us of the cells that are found in the early embryonic development. So essentially, by adding a set of different biochemical cues to the cells called Yamanaka factors, we can create pluripotent stem cells or induced pluripotent stem cells. Yamanaka was rewarded with the Nobel Prize in Physiology and Medicine for this discovery in 2012. Because now we have these stem cells, now we can differentiate them into cells, any cell type that essentially we are interested in using in tissue engineering, including neurons. So we have the cells, and we have the technologies to generate the cells we need for tissue engineering. So what about the biomaterials then? I don't know if you've had the chance to walk around in the STIAS garden lately. It's a beautiful garden, and you can also find this beautiful sculpture there. It was recently put there. It comes from, or essentially it's the root system of a tree that used to grow over there where the new STIAS building is being built, turned upside down. And if you look closely, you can see that there are rocks inside this root system, that are trapped there in a three-dimensional structure. And this reminds me, at least, of how tissues can look like. So the rocks here representing the cells and the root systems, a three-dimensional scaffold that essentially holds these cells in a well-defined structure. And this material that we find in tissue is referred to as the extracellular matrix. And we can see what an explicit function this has if we take an organ, such as a heart, and remove the cells. And this is relatively easy to do, and that's what you see here. And this you can do through a process called decellularization, essentially exposing the organ to detergents or soaps. And what remains here is this biological biomaterial that the cells reside in. And another artist representation of the extracellular matrix is this beautiful painting by David Godsell, showing the neuromuscular junction, where we have a neuron on top here and a muscle cell at the bottom here. And the space in between those cells is completely jammed, cramped full with large, complex molecules. So this extracellular matrix, it's essentially comprised of 2,000 or more different types of molecules that all fill a biological role. This is just a list of the 50 most abundant or so molecules in the extracellular matrix, and it reads a little bit like poetry. Collagen, I'm quite sure you've heard about. There are many types of collagens. And in addition to collagen, we have elastin, fibrillin, fibronectin, laminin, nitrogen, tenacin, thrombospondin, osteopontin, vitronectin, hyaluronan, aggrecan, decorin, bioglycan, perlecan, versican, syndecan, glypican, lumican, osteoglycin. Beautiful, right? And these form the structural framework, the root system, if you like, that keeps the cells organized in a three-dimensional structure. And in addition to the structural role of the extracellular matrix, there are also lots of signaling molecules in there, such as growth factor, which dictates how the cells grow, proliferate, differentiate, and interact. And we also have a lot of molecules that essentially contribute to the constant remodeling, the breakdown and build-up of the extracellular matrix. So this is a material that it's not static. It changes all the time. And the fact that it has such a fundamental important function, we can easily see. If we take extracellular matrix, you can actually buy it off the shelf from suppliers. And we take stem cells and essentially put this in these extracellular matrix materials and provide the right conditions for cells to grow certain growth factors. You can develop something that looks like and behaves like tissues or even organs. So what you see here is some work from the Knoblauch lab in Vienna illustrating brain organoids, mini brains. They typically don't grow larger than this for various reasons. They have, for example, no vasculature, which prevents further growth. This material, it's biologically derived. You produce it in rats or mice by inducing tumors that are rich in extracellular matrix. They can look like this, so pretty nasty. And as you can understand, this process of generating these materials is somewhat unethical, or at least it has ethical issues. It's also problematic that it's derived from animals, which makes it very difficult to translate it into patients, essentially, these kind of tissues that you grow in harvested extracellular matrix, because of the risks of disease transmission. For various reasons, these materials are also very hard to handle and process. And since biologically derived, it contains all of these 2,000 different molecules, and many of which we don't even know what they do. And that can also be a problem. And that also relates to this issue, which essentially something that scientists don't like, that it makes the experiments very hard to replicate because there can be variations in batches between these materials. So what me in my group and our colleagues is trying to do is essentially to answer this question. Why can't we make something that is synthetic or at least semi-synthetic that behaves and functions as the biologically derived extracellular matrix for engineering of tissues? So what we do is that we look at this, essentially what is the composition of the extracellular matrix, and then we start to playing around. So this is an actual picture from the lab, to what bits and pieces we can essentially use in this big Lego and see if we can figure out ways to put it together into something that essentially makes sense from a biological perspective. And we like to take a pretty reductionistic approach. So typically starting with just one of these, and then we add different pieces to increase the complexity rather than the other way around. And we like hyaluronan. It's a very abundant molecule in the extracellular matrix. We also like it because we can do chemistry on it. So this is potentially, I think, the most technical slide I will show today. So this green thing here represents this extracellular matrix molecule, hyaluronan. So it's a carbohydrate. And we modify it chemically to attach something we call a cyclooctene. That's the ring-like structure you see there. And we do this because then we can combine it with another molecule that has these purple triangles here. These are A sites attached to another synthetic molecule called polyethylene glycol. And this is a beautiful chemistry. I'm pretty sure you're familiar with superglue. So essentially what we have here is kind of a biological version of a superglue. So we can take any of these components, throw them together, and we get a solid material. So essentially these purple things react with the ring-like structures through a reaction called a copper-free click reaction. It's a beautiful chemistry that was invented by Caroline Bertozzi, and she got the Nobel Prize in Chemistry for this invention in 2022. And the neat thing here is also that we can do this chemistry in the presence of cells. So we can take these superglue components, put the cells in there, and they will be trapped very much like the rocks in the root system. But still this material behaves and looks quite much like water, right? Cells like, or they are dependent on an aqueous environment. These materials trap water. They're still solid. You can take them up, but they are comprised of roughly 98% water. So that's a good thing, but it's very much like a blank slate to the cells. They can recognize hyaluronan, but there's nothing else of these many other biological cues that the extracellular matrix provide. So then we go back to this list of different components and try to see which of these should we include in order to provoke a specific biological response to the cells. And for example, if you want cells to attach to your hydrogel, then including fibronectin or laminin could be a good thing. If you want the cells to be able to remodel or degrade your hydrogel, then something that can be recognized by these so-called matrix metalloproteinases, MMPs, could be a good thing to include. But we don't want to use these complex, super large proteins that has to be extracted from an organism. Instead, we go to our chemists that uses this beautiful device. This is a microarray-assisted solid phase peptide synthesizer that you see here. Essentially, it's a device that you can program to make the molecules that resembles these extracellular matrix proteins. But we make smaller, more robust versions of them, and also attach these purple triangles to them so we can integrate them in our materials. So here we can make small versions of fibronectin, laminin, or things that are recognized by the cells or proteins that the cells produce. So we essentially build ourselves a library of different components that essentially can mimic the extracellular matrix. And then we do this again. We start to try to put this together. We throw in the cells and we observe what happens. Of course, starting with an hypothesis, if we include this piece, we think this is going to happen. And these are some examples of work that we've done recently. And if you look closely at this picture here, it's not really a picture, it's actually a video. And you can see that the thing is moving. So these were originally skin cells. They were reprogrammed into human-induced pluripotent stem cells and then differentiated into cardiomyocytes. So cardiac cells that, when encapsulated in these materials, start to organize and form cardiac tissue. And as such, it starts beating synchronously. We can also take neuronal cells, also originally skin cells. It's been reprogrammed and start to see if we can grow something that looks like and behave like neuronal tissues. And more recently, we've been interested in cancer. And you may ask, why cancer? Why do you want to grow cancer in the lab? Don't we have sufficient problems with cancer as is growing in our bodies? And yes, that's true. We have those issues, and that's exactly why we want to grow it in the lab also. And one of the main issues that we are trying to deal with is this issue. Essentially that many of the drugs that are being developed, or that the pharma companies are trying to develop, never become viable drugs. So when you develop a drug, you have to go through a very long process in making sure that they do what they're supposed to do without harming the patient. And when you have confirmed that, you can start with clinical trials in patients. And then it turns out that many of these drugs doesn't really do what they're supposed to do. Actually, as much as 90% of the drug candidates fail in clinical trials, and that's extremely expensive problem. And it's also a problem, of course, for the patients waiting for these drugs to be developed. And one of the main reasons for this issue is that we are not rats. And when trying out a drug candidate on a rat, you don't get the same response as you would do if you try it on a human, but we can't really try them on human before animal trials. So what we are trying to address here essentially is providing better physiologically relevant human disease models, which, for example, can enable a faster development of the drugs by trying them on something that looks like and behaves like human cancer tissues in the case of development of a cancer drug, but also to do more fundamental biology, essentially. But this is non-trivial, we've realized, or actually we had an idea of that before we started, because tumors are not just cancer cells, right? If we look closely on a tumor, in addition to cancer cells, there are immune cells there. There are blood vessels, meaning that we have endothelial cells, there we have pericytes, smooth muscle cells, and there are also these fibroblasts, the stromal cells. And all of these and many other cells contribute, we think, to cancer progression. So the question we had to start addressing, essentially, how can we develop something that, at least in a systematic way, replicates the spatial complexity of a tumor? So put things together in a rational way that allows us to figure out how things essentially communicate in these tumor microenvironments. And this is where the biofabrication strategies comes in. So these are essentially technologies to bring cells and materials together in a rational way. You may be familiar with 3D printing. So this is a conventional 3D printer. It works with thermoplastics, so plastics that melt when heated. And you can extrude them when they are heated because they are fluid. And then they cool down, and you essentially just program this extruder to move in a specific pattern, generating an ocelot. We can't use that kind of 3D printer because cells don't tolerate heating, and we're not interested in using thermoplastic. We must use excellular matrix mimicking materials. So we use a technology called 3D bioprinting instead. It's more or less the same thing. You have a robotic arm moving according to an instruction that we program, and it extrudes our materials. But this puts a lot of design constraints on the materials, because the materials must be possible to extrude through a syringe when you apply pressure, and it should solidify on the other end. And all of this without killing the cells in the process. So that's a lot of fiddling around with the materials, with the chemistries involved, as well as seeing what happens to the cells. But we are starting to get there at least, and what you see here is a tumor. I don't know if it's possible to dim the light a little bit to see the colors a bit better. The purple heart here. It's better with these. Yeah, now it's easier to see. The purple heart here is the tumor. So inside this purple heart we have breast cancer cells growing. And it's hard to see them actually because they're not proliferating very much. But they are there, and around these we've bioprinted some fibroblasts, so these stromal cells that we think play a large role in cancer development, and this is also supported by literature. So many has observed this effect that there is a strong crosstalk between these two cell types in cancer progression. We can see that the cancer cell sits there inside this tumor microenvironment that we've created. The fibroblasts are there, but they're not doing very much. And the reason in this specific case is that we've excluded a component of the extracellular matrix mimicking material that prevents the cells from degrading or remodeling the materials. And we did this because we were interested in seeing what impact those specific enzymes had on the cancer cells, especially in the crosstalk between the fibroblast and the tumor cells. So when we essentially just tweak the material a little bit by including one component that allows the fibroblasts to start degrading the cells, we see that the cancer cells start growing quite a lot. So we get a progression of the cancer, and this is just related to how they respond to difference in the extracellular matrix, essentially. So this enables us to, in a systematic way, see what factors can influence tumor progression, and also start mapping out the crosstalk between different cell types in the tumor microenvironment. As you understand, this is a very reductionistic approach, adding piece by piece to a puzzle in order to elucidate what factors influences in tissue maturation and growth. From my humble perspective, I think this is not necessarily the easiest way to creating tissues in the lab, but it's indeed a very interesting approach. We learn a lot, and we may eventually get there. But essentially, paraphrasing Richard Feynman here, what I cannot create, I do not understand. So it helps us to understand fundamental biology, and we learn very much about the biomaterials and the biofabrication processes. But I will also like to tell you a slightly different story on how we can work on this in order to create materials that can enables us to also promote a regenerative pathway. So starting with cells, biomaterials, and biofabrication strategies, similar to those that we've developed for making cancer models, we try to put these materials in a biological context, essentially in tissues, in order to promote healing. And we have, in my group, focused on wounds, and specifically burns, primarily because this is such a huge medical problems. And especially in this part of the world, and especially among women and children. And this is related to cooking over open fires, which unfortunately results in a tremendous numbers of burns and severe burns in children. And they can also results in high mortalities, of course. And we've started many years ago working with a plastic surgeon in Nairobi, Emmanuel Elisom. And that's the gentleman over here, and these are my two PhD students, Petter Sevlier and Morten Skog. So they recognized the problems they were facing when dealing with these children with burns, that many of them didn't get adequate care, and that resulted in significant scarring. And the reason why you don't get care is, of course, that this is extremely expensive. Many of the methods that are currently used in treatment of burns involves transplantation or use of advanced dressings, such as those you can obtain from either pig skin or from deceased people. And either for religious, economic, or cultural reasons, you can't really use that in many of these patients. And I will show you some pictures in the following slides that some of you might find disturbing. And I apologize for this, but I think it's important to just understand the scope of the problem. So this girl, she was suffered burns after she was raped and thrown into fire. And she survived with significant scarring as a result. The scars on the inside we don't know much about, but on the outside it really impaired her life and life quality. And she had troubles eating solid food, for example, or moving, in addition to the aesthetic problems. Emmanuel could, after significant surgery, at least reduce some of the contraction. But this is what happens when you don't get appropriate care, right? And what Petter and Morten realized, that there is need for skin substitute that are available to this group of vulnerable patients. They are cheap and readily available. So they started looking into different extracellular matrix mimicking materials that could be made at scale, essentially. And the most abundant biopolymer on this planet is cellulose. So why not use cellulose? That should be cheap. So they developed a strategy, a biofabrication strategy for making these skin substitute from nanocellulose. And if you look in the electron microscope, they look very similar to the nanostructure of skin, actually, to the collagen matrix that you find in skin. And this skin substitute can be applied to a wound, and they adhere to the wound bed and sits there until the skin has healed, and then they fall off. So this is what can look like when you treat a burn patient. This is a child with different types of burns or different thicknesses of the burn, partial thickness and mid-dermal partial thickness burns. It's very painful. But when you apply these skin substitutes, the pain typically decreases significantly because of the capping of nerve ends, we think. And after just a few hours, the dressings adhere to the wound, and then they stay there until it falls off. So it protects the wound for this period of time. And we also think that it reduces scarring. But the main purpose is here essentially just to promote healing, essentially. Not make it worse, at least. It turned out that these dressings was also quite effective on so-called chronic wound or non-healing wound. So this was one of the cases where this patient had this wound for 20 years, going to the doctor a couple of times a week to get her dressings changed, and with these skin substitutes, these wounds healed in 7 months, which was pretty fantastic. So it can provide a cost-effective, healing-conducive microenvironment, but it doesn't really solve the problems with scarring. So together with colleagues, we started to think about how can we deal with that problem. And we tried a strategy where we harvested skin cells from patients. And in order to rapidly expand skin cells, we needed to figure out a way to do this, because if you have a burn, you can't just wait weeks and weeks in order to expand the cells. Cells don't really grow that fast. So we identified these excellular matrix-based microbeads. They look like this on the microscope, primarily made of collagen. And these are also water swollen. So we see the cells on these beads, and we transfer them to a device called a bioreactor. It's essentially a device that provides ideal conditions for cells to grow. So providing nutrients and oxygen and the correct temperature. So nothing really complicated. And after 3 days, we can harvest these beads, and we see that these beads now are essentially covered with skin cells. We refer to these as skin tissue units. And in order to be able to transplant them, we need to encapsulate them in a material. So we take our super glue, extracellular matrix mimicking materials, and combine them with the bead, and then we end up with a material that looks like this. So a skin cream in its own right. And we can process these in different ways for transplantation, either that we bioprint them into construct that we can essentially put in a wound, or that we essentially extrude them directly into the wound. And when we do this, we can see that over time, these beads start to degrade. So what you see here is something called histology, and we can still see the beads here, but we also see new tissues being formed here, which doesn't really look like scar tissue. And we can also see, if we start counting the cells that we transplanted, that they are initially having a bit of a struggle, but then over time, they keep on proliferating and growing. And that's a good thing in this context. And we can also see, if we look at certain molecules in these images, that we have formation of blood vessels, which is also a good thing. And now I'm bombarding you with data, because that's something we as scientists like, right? When looking for what extracellular matrix proteins there are in there, we can do this by both looking at the genetic profiles and what essentially components are actually being produced. By using various staining techniques, we can see that we have many of those molecules that we need for a functional tissue architecture, to us indicating that we are on the track of being able to induce regeneration in these wounds. And this is work still in progress. And I didn't do much of this work. It was my team members in my group, the Laboratory of Molecular Materials. and in collaboration with, among others, the Laboratory of Experimental Plastic Surgery. And I, of course, also would like to acknowledge all our funders. And thank you for listening. If you're interested in knowing more about our research, you have the QR code to our webpage here.
Thank you, Daniel. It was remarkable. The extracellular matrix is, I think, even more fascinating than the movie.
Yeah.
Which you know, for someone like me, that's entirely new learning, and you made it accessible, and for that we thank you. But I think also just showing some of the new approaches in your field, really path-breaking and exciting, and wonderful to listen to you. So I wanted to invite, we've got some good time. Thank you for leaving a lot of time for us for questions. I see several hands already, so we'll start with Catherine, Ilan, and Kepa. So.
Thank you so much for your lecture. It's fascinating. So, as a humble surgeon who removes breast cancer and treats people with burn wounds, the potential is amazing. But maybe just because we didn't learn that the extracellular matrix was so important, and in terms of like signaling and turning on cells that were already differentiated to do things, we always learned about growth factors. So is that some of the things that are in the ECM milieu, and you're trying to figure out which are important? But I guess the question is, in terms of the timing of when to turn things on and off, especially if it's preventing growth like cancer or encouraging growth like regeneration of new skin, how does all that play? Because right now it looks like you just put the right things in the pot and it'll do the right thing. But what about in terms of timing? Thank you.
That's a very good question, and there is no answer to that. At least with our current knowledge, we don't know the timing of growth factors. There's been plenty of work in tissue engineering to try to add growth factors, different growth factors, and playing around with the timing. But it's complicated, these regenerative pathways, and also the fact when you add a growth factor to an injury site, you also have degradation. So other proteases that degrade these growth factors. So we can only be humble and conclude that it's complicated, and we need to know more about this and how it influences regeneration. But I think this is one way to do it, and essentially try to provide a healing conducive environment, and then we can do proteomics, for example, at different time points during healing to learn more about this. But extensive amount of research has been devoted to this question.
Thank you, Daniel. It's really a wonderful talk and fascinating. So much stuff to try to understand in that. In some ways, I want to follow on Catherine's question, but slightly backwards maybe. Because of the interest in the interest in the cancer cells and the cancer growth, what is it that, in a sense, if you were to think of this starting backwards, that is, you've got this extracellular matrix, you've got the potential, if you will, for growth, for continued growth. And the cancer, the problem is that it does continue. So what would help you see what stops that continuation? Can one look at that in this context?
Yeah, that's a very good question. And when you make these kind of simplistic model, the cancer will stop growing in most cases. But it depends a bit on the materials you put them in and the context and what you feed them with. For example, if we make a material that doesn't degrade, as we showed here, they are essentially stuck in there. So there's no room for them to start continuing expanding. They need to be able to degrade their microenvironment in order to be able to expand. And eventually, in these kind of models, they will form something called spheroids, which essentially is a structure a couple of 100 microns in size, and then they will stop growing, essentially. And the reason for that is because they are starving the cells inside here. There is no vasculature in these models that provides the cells with the nutrient and oxygen they need. So in order to increase the complexity of these models, that's something we're trying to figure out ways to essentially create vasculature that are perfusible. And that's also a very interesting bioengineering challenge. But if you just provide the right conditions, they will continue to grow. I mean, you can form more and more of these spheroids eventually, and it will be just crammed in your tissue culture plate eventually.
Thank you.
Thanks also from my side. Wonderful, interesting, impressive work. Couple of questions. One is, how do you deal with tissue specificity? Because I guess that the extracellular matrix is also tissue dependent. Like, depending on the tissue that you are working with, you need to change, let's say, the ingredients that you have there so that it works. I suppose that that's something, there's a challenge for you as well. And the second question is, when you were speaking about connective tissue, the boundaries between connective tissue and the actual extracellular matrix, in the sense that the connective tissue is exactly what connects, the different cells are in our body. How do you draw the boundary? I suppose that connective tissue is also responsible for the generation of that matrix. How do you kind of tell the fibroblasts in cancer, for example? I mean, fibers, they are connective tissue. It's connective tissue. So I suppose that they also sequester kind of fibroblasts to do their job for the cancer. I suppose that there is an issue there with sequestering those type of cells to create their own matrix. Well, just
to start with, regarding tissue specificity there, that's absolutely true. Different cells require or respond to different materials properties. So that's, of course, something we like to play around with, and for example, by just changing the stiffness of these materials. So brain tissue, if you want to culture neurons, you want something that is really soft, typically. And then we tune down the stiffness of the material. If you want to go for something else, like more connected tissue, you want to kind of increase the stiffness, and that's also possible to do. And then, of course, all these other components that needs to go in there can also determine how the cells respond to these materials. Neurons, for example, they want laminin, so then we can put in some of these molecules that mimics laminins. Or sometimes we put in even entire large laminin proteins in there. And regarding, I mean, what happens here with the essentially production of extracellular matrix by the proteins, and or how they remodel and how that relate to connected tissue. I mean, there are so many aspects of this, but I mean, of course, when we culture cells in these materials over time, they start producing their own extracellular matrix, and especially true if we have fibroblasts there, which essentially are heavily involved in production of extracellular matrix. But it also depends on materials properties. So it's a constant crosstalk between the cells and the materials, their microenvironment, just as we respond to whatever is around them.
Thanks. We had Nigel.
Yeah, fabulous talk. Thank you. I'd just like to come back to the old axolotl. I mean, so what's that trick then? I mean, it's interesting to me. Do the close phylogenetic cousins of axolotls also able to do this trick, or what is it that system has that gives them almost a complete capability to regenerate across quite a wide range of cellular types?
Yeah, fascinating, and also something that is not fully understood, and that's hence the research ongoing here. What is known so that's is that they can control the inflammatory response to a large extent, and because that's something that controls the wound healing process. So they can downregulate the inflammatory response, and they can also ensure that you have a process that is similar to the formation of organoids. Essentially that you have these stem cells residing or being recruited to the injury site and starting producing new tissues. But the exact mechanisms surrounding this is still ongoing research.
Thanks. Next up was Ben.
Thanks so much. Thanks, Daniel. Wonderful work. So, and as a physiologist, I'm of course super excited to see the role of physiology. If we think 25 years down the line of the human genome sequencing, we see the role of the functional networks to actually impart cell function. So this is really wonderful to see. So I have two questions, and they're both related to complexity. So do you think if we look at for example, this redifferentiation of fibroblasts into neuronal cells and human-derived, how far are we with this kind of a single system, single cell type, to do this in some kind of routine, semi-diagnostic or personalized approach? And then my second question, more to the more complex, like embryogenic, microenvironment, is it maybe too complex generally? Is the human mammalian development too complex to recreate certain microenvironments? So how do you see it? These two kind of what's going on in the field?
Yeah, so relating to the first question, I think this is something that we start to see appear, essentially personalized disease models being created in the labs, where patients with specific genetic conditions can have essentially their own personalized disease model being produced in a laboratory in order to test out how they respond to specific drugs. So this is an amazing development, and also the fact that you can start using organoids in order, more generic organoids. You can take a biopsy, for example, from a cancer, from a glioblastoma, insert it into a brain tumor organoid, and see how that specific cancer respond to treatment. I think this is the potential, has a very large potential for personalized medicine, and we will see a lot more of that. And regarding the complexity here, I agree that this reductionistic approach, it's going to be very challenging to put together entire tissues and organs using this, but we can learn a lot from this process, essentially what bits and pieces that does essentially specific biological things, but the entire complexity of biology is so tremendous. And we need to work on that first, I think, to try to figure out the biology, and then we can, in parallel, perhaps do the bioengineering work here. But as I see it, hijacking biology a bit, combine the material cells, put them in a biological context. This is where I think we can start to, at least as of now, really do progress towards essentially regenerative pathways instead of scoring.
Thanks so much.
Thank you. Bruce over here was next.
Yeah, thanks very much. This was really interesting. The frame of reference for all of your examples was bringing the body back to full normal human health. And I can't help but think, and maybe this is my philosopher brain going off in weird directions, but I can't help but think that there are other frames of reference. I'm thinking transhuman, post-human kind of questions. There are people who want to grow tails. There are people who want to have body modifications of all sorts. There are, in other words, alterations to the human form rather than restorals of the human form. And I'm wondering, I know you might just say this is out of beyond your pay grade or out of your... But I'd really be interested in hearing what you have to say about this, because it seems to me it's not just about restoring, or potentially not just about restoring to full health. It could be about alteration as well. And what do you see in that?
Oh, that's an interesting philosophical question. First and foremost, I can focus on solving some immediate medical problems, but looking beyond is very exciting. We have, together with collaborators, looked at integrating electronics in these materials to essentially being able to have cell and biocompatible electronic interface with cells. So we and others are looking into those possibilities as well, because it's exciting, and it can also help us to, in addition to looking beyond health, actually solve some medical problems. So yeah, there are also exciting development on that side, I would say.
Okay, fascinating. Next up with Chepiso.
Thank you very much for your talk, Daniel. I just wanted to find out, will this kind of models, in terms of the cancer models, work for cancers like leukemia? Because these are solid matrices.
Yeah. We're really interested in solid tumors in this case here. Yeah. So looking at essentially recreating the tumor microenvironment in solid tumors, and primarily then in breast cancers is what we've been focusing on.
Okay, thanks. Next was Alex.
Okay. Thanks, Daniel. Really very exciting presentation. You said that part of your main motivation is to create cancer models that could be used in drug trials and development. And what would be, how different would this model be from what we already know of, say, breast cancers currently? And how does the starting base for for the cancer model that you generate, how does that relate to the various differences that there might be with breast cancers, and there are different types, and how would your starting point relate to all those variations that we see within breast cancers?
Yeah, so that's a very good point. I didn't go into that really, what the limitations of current models are. So as of today, when studying breast cancers, you use also human cell models to a large extent. Then you culture the cells on tissue culture plastics, so essentially a piece of plastic. The cells readily grow there, typically, proliferate and expand, but it's a completely different environment compared to tissues. And the cells consequently respond very differently. But it's a very good tool, has been a very good tool for fundamental cell biology, for development of drugs, and still is, and widely used. But it has these limitations that the cells respond very differently to what they do in tissues. The next level of complexity are essentially animal models. And then you drastically increase the complexity. So there is no really thing in between here. So in addition to increasing the complexity, it's also non-human, right? So what we try to do, and our colleagues working in this field essentially, is to provide a model that can, with respect to complexity, be somewhere in between two-dimensional cell culture and animal models, but also provide the human context. So that was the answer to the first question. And the second question, that was?
Your starting point for the cell, for the model.
Yeah.
How does it relate to the various cancer types, even when you think of breast cancer?
Yeah, I mean, cancer is such a diverse disease. I mean, it's many diseases, essentially, just between different types of breast cancer, for example. Each and every cancer is probably unique, right? But in the best scenario, we can have a biopsy from a patient and essentially make their own breast cancer model or cancer model and use that to try out which drugs is the most efficient on that specific patients. So we primarily worked on breast cancer. We also looked a bit into neuroblastoma and developed models for that, which is brain cancer, as well as liver cancer, and using similar strategies here. As a platform, this strategy is fairly flexible. I mean, as I mentioned, we can tweak the materials properties in a more like Lego-like fashion to essentially optimize this for different types of disease models.
Okay, thanks. Alex, we had a person with the blue jersey over here.
In your model, using the cellulose seeded with skin cells, what is the ultimate barrier? Is it the matrix, the cellulose, or are the cells functioning and they fulfill that role of the biological barrier for reinfection?
You mean when we try to or transplanted these beads into humans or into wounds, you mean?
Yes.
Yeah. Yeah. So I mean, here we only have one cell type to start with. It's fibroblast that we start with, whereas skin is comprised of many different types of cell types. My colleagues have investigated the impact of transplanting beads with different cell types. They didn't see much of a difference, if also including keratinocytes, for example, on the healing process. So in a way, we don't really know what the impact of increasing the complexity of these tissue constructs are essentially. And this is still work in progress, so we will see what essentially the main barriers will be when it comes to, I mean, just inducing healing. Yeah. But then, of course, in developing a treatment, there are many barriers, but that's of a different kind, more regulatory.
Thank you.
Thank you. Over there with the blue shirt, yes.
Yeah, thanks for a very interesting talk. My name is Barend. I think your subject is too complex for me to ask specific questions. So I've been thinking about the environment in which you operate. And I'm specifically thinking about the skills that you require, the team that you build, and the interfaces that you have to be successful in this environment. And I'd like to touch on sort of commercialization of this type of technology as well, if you want.
Yeah, I mean, this is very interdisciplinary research, and it's hard for a single lab to do this. So it needs to be done in collaboration, and we work closely with multiple different groups in and around Sweden primarily, and also together with clinicians. And I think especially when having some sort of translational aspects in mind, that's a very important part, because we can spend years of developing a super sophisticated materials, and the first thing they do is to essentially cut it into pieces and chop it up. And if we knew that they wanted to do that, we would probably have done it in a different way to start with. So talking with the clinicians, how they work and what they want to, what they need essentially. That's a good start. So having an interface where I actually can meet is very important. And at Linköping University, all the PhDs in life science and life science technologies are enrolled in a graduate school that is interdisciplinary. So they actually get the chance to meet already at the PhD level, which I think is good, because many of those ideas comes from the PhDs just bouncing ideas or drinking coffee. So having opportunities to meet and discuss is good, and involving people and creating kind of a collaborative environment. But then also in the group, you need a critical mass of people with different competences in organic chemistry, in biochemistry, in bioengineering, at least in my group, in material science also. So that's one aspect. And then when it comes to commercialization, that's also a separate process. When it comes to medical devices, that's also quite regulatory complex, and the EU, it has become even more complex since the adoption of the new medical device regulatory framework. But having close contact with the innovation office is something that we have seen is very helpful. Getting some funding for patents and help to write patents, and from that process essentially knowing how to deal with starting a company and getting people involved in that. It's a lot of work, but that's also quite interesting and fun.
Great. Thank you, Bernd. So Kepa wanted to bring up another point.
It's so fascinating that I wanted to jump several times in there. but one of the questions that came up, like with some of the issues that people were putting on the table, was, and could be one of the limitations in terms of, in our body there are many cells that move around. It's not just the liquid, like blood, but it's also the part that has to do with the immune system. There's patrolling everywhere. And when you responded saying that one of the keys to regeneration could be to control the inflammatory response, is precisely those cells that move, right? And it could be a bit like, can you elaborate a bit more on that? Because it was very...
I mean, it's a super exciting research topic that is also a lot of things happening now, and essentially biomaterials that control macrophage polarization, for example. If it goes through a pro-inflammatory or pathway, or macrophages that essentially promote healing. So controlling macrophage response is probably one of the key factors to regeneration. With or without cells in the biomaterials, but there are strategies being explored in that direction. And we are also, of course, trying to look at what happens when we put immune cells in these materials.
Thanks. I will catch you around for further questions.
All right, thank you. Any final comments and questions? Yes, please.
Hi. Hello. Thank you for a very interesting talk. I hopefully have 2 questions. The second will depend on the answer of the first.
Okay.
I just want to find out, so we saw the cellulose almost plaster to help with skin regeneration or just healing, and then the beads with the skin cells, and I just want to find out the goal of these beads. So you saw the skin cells that are on these beads, they do, you give them enough ECM proteins or growth factors, enough oomph, for lack of a better word, to grow and develop. Is the ultimate goal for them to just attach to each other and try and create this barrier on their own, or also involve the individual's skin cells, blood supply, whatever, to try and fix this wound in the skin?
So the idea here with these beads is essentially to be able to rapidly expand cells, first and foremost, but also in a way that we don't have to kind of detach the beads from this, or the cells from the support. Because here we can essentially make a living material that we can implant. There are current strategies for expanding skin cells. So essentially you take skin cells from if you have a patient with burns, keratinocytes primarily, and then you culture them into thin sheets that you can harvest and then essentially put on the wound. It takes a very long time, several weeks, and you have to essentially chemically treat these cells multiple times, which essentially make them lose a bit of their kind of abilities to function. And we try to circumvent that essentially, to make this process more rapid and to essentially provide a better environment for these cells after transplantation, which we think also, and what our data indicate, can provide a better regenerative microenvironment than for essentially reducing scarring. We also saw that after implantation there appears to be ingrowth of blood vessels, which is of course important then for creating a viable tissue. I don't know if that was the answer to your question.
No, that was. Thank you very much. And then just my second question is, do you think a further goal of this research very far down the line, maybe from your research group or just in this research in general, would be to try and find or create these beads or different biomaterials to work together in layers or something to try and involve the human body in healing of these different tissues that are affected? So if you have a really deep scar and maybe some fat or muscle is affected, other blood vessels, to try and involve the body in this more intense repair.
Yeah, and I don't think it's necessarily too far in the distance, in the future. Actually, we are already in preclinical trials with these materials in large animal models. Of course, the step to clinical trials is long and complicated, but that's just us, and there are many research groups moving in this direction. So I think we will see interesting progress, actually, towards regenerative pathways.
Thank you.
Thank you. Any last hands? So, yes.
Hi, Daniel. Thank you so much. That was incredible. For someone that doesn't know anything about biology, it was very interesting. My question is also about the biosynthetic nanocellulose. Because from how you describe it, it's so much more effective and efficient than other forms of treatment for burn wounds. So my question is, how is it more cost effective if, from what I'm gathering, it has nothing but pros?
So we know it's roughly as effective as porcine skin. So not necessarily better, but instead of harvesting skin from an animal, we can make this in the lab. Essentially, this specific nanocellulose is produced by bacteria. So we grow them in large tanks, and they produces this as a thin film that can then be further processed into to these skin substitutes. So compared to other materials, this is cheaper. But it's also cheaper in the way that you apply them once, and then they sit there. Whereas many other strategies, you have to constantly replace whatever you put on the wound. So even though the initial cost can be higher than certain materials, the total cost for treatment is tend to be lower. But then also in ongoing work in my lab, or with colleagues at the Luleå Technical University, also in Sweden, we've developed a strategy to produce identical materials but from trees instead. And we have plenty of trees in Sweden, Canada as well, right? So this is essentially to be able to scale up the production of these materials and make them even cheaper and more accessible.
Great. Yes, at the back, please.
Sorry, this doesn't have much to do with biology. But I noticed you had some very nice images of your cells in your presentation. Do you know anything about the imaging system you used to get these images?
It depends on which images you mean here, but it's we use a Clariostar confocal microscope.
Just like a normal confocal microscope.
Yeah, so these are mostly conventional confocal microscopy when it comes to the imaging of the cells and tissues. Yeah.
Thank you.
Thank you. That's an important part of the presentation, yes.
Yeah.
So I'm sure there are more questions, but there is an invitation to ask them to Daniel over a glass of something to drink and something to eat. Ben, I think you mentioned we're 25 years into the mapping of the human genome, and we've come a tremendous far way, but we've only just started, it seems, as well. And I'm pretty sure in another 25 years, when we have you back here, Daniel, maybe to talk about your career of discovery over this period, it will be a completely incredible picture as it already is. So thank you for all those questions, participating in the conversation. Daniel, thank you very much. Thank you for being at STIAS with us. This Daniel is returning to Sweden soon. So thank you for your presence. Thank you for the wonderful presentation.
Thank you. And thank you for all the great questions. Very interesting discussion.